The nervous system is amazingly complex in that each part of it, whether it's from the back of the eye sending information into the brain, or the brain sending it down to the spinal cord for movement, or one part of the brain sending it up to another, each of those parts of the nervous system have these nerve cells or neurons that receive the information and send it and process it. But they're surrounded by support cells called glia that give them nutrients, that take away toxins, that make sure they survive well, and frequently surrounded by other cells in some areas of the central nervous system called oligodendroglia that essentially wrap a rubber-like compound around those arm-like axons and let them transmit information correctly. So no matter where in the nervous system we are, there's a nerve cell, support cells, and these wrapping cells. And in nervous system disease, different types of nerve cells or glia degenerate or are injured. At HSCI, we've broken into 5 working groups in the nervous system, dealing with blindness and diseases of the retina, the inner ear leading to deafness, the motor system leading to diseases like ALS or Lou Gehrig's disease or spinal cord injury, the system that has to do with Parkinson's disease, and studying those support cells themselves, glial cells that are involved in multiple sclerosis and many types of cerebral palsy. In the HSCI Nervous System Diseases program, a group of 5 labs have come together from across Harvard University to bring their technological and scientific expertise together, and we've uncovered a whole set of molecular recipes for these cells, to the point where we can actually activate the birth of entirely new neurons of the right type in the brains of laboratory animals.
Yeah, I'm going to start there.
A premier example of how developmental or stem cell biology can contribute to disease therapies in the laboratory dish rather than by making replacement cells is the work by Kevin Egan and Lee Rubin in the HSCI Nervous System Diseases program, where over the last few years around the world, enough information has amassed that they can build billions and billions of exactly the kind of neuron that dies in the spinal cord in Lou Gehrig's disease or ALS. That happens to be exactly the kind of nerve cell that also dies in a fatal childhood disorder called spinal muscular atrophy. And by studying this one kind of nerve cell, there's opportunity to find therapeutics, to find drugs, to find molecules that could reverse the childhood disease potentially entirely and 1/2 of the ALS disease. Our nervous system diseases program at HSCI has taken 2 large-scale approaches to collaboration. One is that we have such a depth and breadth of experts across the Harvard system that there are a number of newly formed internal collaborations between people who might just be 3 or 4 miles away at different corners of the university, but who've never met or certainly never interacted, though their science not only is complementary, it's necessary to leverage something to a whole higher level.
Developmentally. Yeah, so it's not level specification, but sort of early progenitor.
Another kind of collaboration that we've been very interested in at HSCI is bringing Harvard investigators together with those elsewhere in the world. And to do that, we've started a series a few years ago of what we term think tanks. The think tanks we've run for the first 2 years focused on Parkinson's disease, and that was really successful, getting us to some biology where we think HSCI investigators and resources can contribute immensely to the field, both here and with international collaborations. And then last year, we moved to the idea of brain repair and therapeutics in ALS and spinal cord injury. Many in the basic science world have traditionally thought of their work as separate from work that could ultimately help people. And HSCI has provided a vehicle by which very basic developmental biologists have been able to see their work as relevant to human disease, to understanding what goes wrong in disease, how you might fix it. And they don't need to want to do that in their own lab. They can find partners that take their basic ideas and their basic discoveries and bring them through that HSCI pipeline to get to therapies and to get to ultimate restoration of function in the nervous system.